Welcome to this video on cell structure and function. Today, we'll be exploring the microscopic world of cells. By the end of this lesson, you will be able to prepare temporary slides for viewing under a light microscope, draw cells accurately, calculate magnification and actual size, understand the difference between magnification and resolution, and identify the structure and function of key eukaryotic organelles. Let's begin by learning how to prepare a temporary mount, a fundamental skill in biology. Our first learning outcome is to be able to make temporary preparations of cellular material suitable for viewing with a light microscope. So, what exactly is a temporary mount? It's a preparation of a specimen on a microscope slide, typically using water or a stain, and it's designed for short-term observation. It's a quick and easy way to look at living or freshly collected cells. Let's walk through the steps to create one. First, you need to obtain your specimen. Ideal choices are thin sections of tissues, like the epidermis from an onion or cells scraped from the inside of your cheek. The key is that the specimen must be thin enough for light to pass through it. Next, you'll place this thin specimen in the very center of a clean glass slide. You can use forceps or a needle to handle it carefully. The third step is to add a drop of water, or a stain. Water is used to keep the specimen hydrated and prevent it from drying out. Stains, on the other hand, are used to highlight specific structures within the cells, making them easier to see. For example, iodine solution stains starch and enhances contrast, while methylene blue stains nuclei. Now for a crucial step, adding the cover slip. You must lower a small, thin cover slip gently over the specimen. The best technique is to start from an angle and slowly lower it down. This helps to prevent trapping air bubbles, which can obstruct your view. After the cover slip is in place, you might have some excess liquid around the edges. Use a piece of filter paper or tissue to soak this up. This keeps your microscope stage clean and prevents the slide from slipping. Finally, you're ready to view your slide. Always start with the low-power objective lens, typically 4x or 10x. This gives you a wide field of view, making it easier to locate your specimen. Once you've found it, you can switch to a high-power objective, like 40x, for a more detailed observation. So, to recap, we've learned how to prepare a temporary mount by obtaining a thin specimen, placing it on a slide, adding water or stain, carefully adding a cover slip, removing excess liquid, and finally viewing it under the microscope. Let's take a closer look at why we use stains and some important safety tips. As we mentioned, stains are incredibly useful. They bind to specific components within the cell, making them visible. For instance, iodine solution is used to stain starch, which is abundant in plant cells, and it also enhances the overall contrast of the image. Methylene blue is a common stain for animal cells, as it effectively stains the nucleus. Acetic orsin is another stain, specifically used to visualize DNA and chromosomes, which is very helpful during studies of cell division. Now, a quick word on safety. When working in a lab, safety is always the top priority. First, always handle glass slides and stains with care. Glass can break and cause cuts, and stains can irritate your skin or eyes. Second, it's crucial to dispose of used slides and tissues properly. Follow your lab's specific instructions for waste disposal to ensure a safe and clean environment. By following these guidelines, you can safely and effectively prepare and view your temporary mounts. Now that we know how to prepare a slide, our next learning outcome is to be able to draw cells from microscope slides and photomicrographs. Why do we draw what we see? Drawing is a powerful tool in science. It helps you understand the structure, organization, and relative sizes of organelles. It also encourages you to develop your observation skills and pay close attention to detail and proportion. It's a way of actively engaging with the specimen. To ensure your drawings are clear and accurate, there are some important guidelines to follow. First, always use a sharp pencil. This allows you to draw clear, fine lines. Second, avoid sketching. Each line should be single and unbroken. Third, maintain correct proportions. Your drawing should realistically represent the size and shape of the structures you observe. Fourth, do not use shading or coloring. Instead, use label lines to indicate different structures. Fifth, your label should be straight, horizontal, and drawn with a ruler, typically on the right side of the drawing. And finally, always add a title that is informative and includes the cell type and the magnification you used. Let's look at an example of a well-drawn animal cell. Here is a drawing of human cheek cells stained with methylene blue and viewed under 400x magnification. Notice how the title is informative and includes the scientific name. The cells are drawn with clear, single lines, and there is no shading. The labels are neatly arranged in a vertical list on the right, with straight lines pointing to the correct structures: the cytoplasm, nucleus, nucleolus, and chromatin. This drawing follows all the guidelines we just discussed. So we've now covered the purpose of drawing in microscopy and the key guidelines for creating accurate and informative biological drawings. Let's continue our discussion on drawing cells by looking at common structures to label in both plant and animal cells. For a plant cell, like an onion epidermis cell, you should be able to identify and label the cell wall, which provides structural support, the cell membrane, which controls what enters and leaves the cell, the cytoplasm, the jelly-like substance filling the cell, the nucleus, which contains the cell's genetic material, and if visible, the large central vacuole, which stores water and maintains turgor pressure. Here is a diagram of onion epidermal cells. You can clearly see the rectangular shape, the thick cell wall, the cell membrane just inside it, the cytoplasm, the nucleus, and the large central vacuole. These are the key features you would look for and label in a plant cell. Now for an animal cell, like a cheek cell, the structures are a bit simpler. You should be able to label the cell membrane, the cytoplasm, and the nucleus. Animal cells do not have a cell wall or a large central vacuole. This diagram of a cheek cell shows these three main components. The outer boundary is the cell membrane, the internal fluid is the cytoplasm, and the large central structure is the nucleus. Sometimes, you might also be able to see mitochondria, which are the powerhouses of the cell. So, we've now identified the common structures to label in both plant and animal cells, which is essential for accurate biological drawings. Let's summarize the process of observing and drawing cells, and then discuss some tips for drawing from photomicrographs. First, always focus your microscope using the low-power objective, and then switch to high power for detailed viewing. Second, observe the structure carefully, paying attention to the outline and position of all the components. Third, sketch the shape of the whole cell first, and then add individual organelles if they are clearly visible. And fourth, add your labels using a ruler to draw straight lines. Now, what if you're asked to draw from a photomicrograph, which is a printed microscope image? The key here is to draw what you observe, not just copy the image. You should trace the outline of the structures you see, but don't include unnecessary background details. The goal is to create a simplified, clear representation. Also, make sure your drawing is large enough, at least 50% of the available space, to allow for detail and clear labeling. By following these tips, you can create high-quality biological drawings from both live observations and printed images. Our third learning outcome is to be able to calculate the magnification of images and the actual sizes of specimens. The key formula for this is magnification equals image size divided by actual size. It's crucial that you use the same units for both the image size and the actual size in your calculation. This formula allows you to determine how much larger the image you see is compared to the real object. Let's work through an example to calculate magnification. Suppose the image of a cell measures 50 millimeters and its actual size is 25 micrometers. First, we need to convert the units. Since 1 millimeter equals 1,000 micrometers, 50 millimeters is 50,000 micrometers. Now we can apply the formula. Magnification equals 50,000 micrometers divided by 25 micrometers, which gives us a magnification of 2,000x. This means the image is 2,000 times larger than the actual cell. You can also use a simple triangle diagram to help you remember the formula and its rearrangements. This triangle has I for image size at the top and A for actual size, and M for magnification at the bottom. To find any variable, simply cover it up. For example, to find magnification, cover M, and you're left with I over A, or image size divided by actual size. This is a great mnemonic device. So we've now learned the key formula for magnification and how to apply it to calculate the magnification of a specimen. In this scene, we'll learn how to calculate the actual size of a specimen and how to use scale bars and micrographs. Using our formula triangle, we can rearrange the formula to solve for actual size. Actual size equals image size divided by magnification. This is incredibly useful when you know the magnification of an image and want to find the real size of an object within it. Let's try an example. Imagine a photomicrograph is magnified 500 times and the image of a nucleus measures 10 millimeters. First, we convert 10 millimeters to 10,000 micrometers. Then we divide the image size by the magnification, 10,000 micrometers divided by 500. This gives us an actual size of 20 micrometers. So the real nucleus is 20 micrometers across. Another common way to determine actual size is by using a scale bar. A scale bar is a line on a micrograph that represents a known actual distance. For example, if a scale bar says 5 micrometers and measures 10 millimeters on the paper, it means that every 10 millimeters on the image represents a real distance of 5 micrometers. You can then use this ratio to calculate the actual size of any other structure in the same image by measuring its length on the paper and setting up a proportion. We've now covered how to calculate actual size using both the magnification formula and scale bars, providing you with two powerful methods for measuring microscopic objects. Let's quickly review the unit conversions you'll need for these calculations. The standard units we use are millimeters, micrometers, and nanometers. 1 millimeter is equal to 1,000 micrometers. 1 micrometer is equal to 1,000 nanometers. And 1 millimeter is equal to 1 million nanometers. Remember these conversions, as they are essential for getting your calculations right. Now let's compare the different types of microscopes you might encounter. We have the light microscope, which uses light to illuminate the specimen. It has a typical magnification of up to 1500x and a resolution of about 200 nanometers. Then there are electron microscopes, which use a beam of electrons. The scanning electron microscope, or SEM, can magnify up to 100,000x and has a resolution of about 10 nanometers. The transmission electron microscope, or TEEM, is even more powerful, with magnifications up to 500,000x and a resolution of about 0.1 nanometers. As you can see, electron microscopes offer significantly higher magnification and resolution. We've now reviewed the key unit conversions and compared the capabilities of light and electron microscopes. Our fourth learning outcome is to be able to use an eyepiece graticule and stage micrometer scale to make measurements. First, let's define our tools. An eyepiece graticule is a small, transparent disc with a scale on it. You insert it into the eyepiece of your microscope. The key thing to remember is that the units on the graticule are arbitrary. They don't have a fixed real-world size. A stage micrometer, on the other hand, is a microscope slide with a very precise scale, usually 1 millimeter long with 100 divisions. This means each division on the stage micrometer is exactly 0.01 millimeters, or 10 micrometers. So why do we need both? Because the graticule has no standard unit, we must calibrate it using the stage micrometer. Calibration is the process of determining what one graticule unit equals in real-world units, like micrometers or millimeters. This value will change depending on the objective lens you are using, so you must calibrate for each magnification. Let's outline the calibration steps. First, place the stage micrometer on the microscope stage. Second, focus under low or medium power. Third, align the graticule and stage micrometer scales so they are parallel, and you can see where their divisions line up. Fourth, count how many graticule units line up with a known length on the micrometer. This is the crucial measurement you need for your calculation. We've now defined the eyepiece graticule and stage micrometer, explained why calibration is necessary, and outlined the steps to perform it. Let's put our calibration knowledge into practice with an example calculation. Suppose you've aligned your scales and found that 100 graticule units line up perfectly with 0.5 millimeters on the stage micrometer. First, convert the micrometer reading to micrometers. 0.5 millimeters is 500 micrometers. Now, to find the value of one graticule unit, you divide the total length in micrometers by the number of graticule units. So 500 micrometers divided by 100 graticule units gives you 5 micrometers per graticule unit. This is your calibration factor for that specific objective lens. Now that you're calibrated, you can measure any specimen. Let's say you remove the stage micrometer. Place your specimen on the stage and observe it. You measure a cell and find it is 20 graticule units long. To find its actual size, you multiply the number of graticule units by your calibration factor. So 20 graticule units times 5 micrometers per unit equals 100 micrometers. The actual size of the cell is 100 micrometers. Remember these two important notes. First, calibration must be repeated for each objective lens. The magnification changes, so the value of one graticule unit will also change. Second, always record the magnification you used for your measurements. This is crucial for others to understand and verify your results. We've now successfully demonstrated how to calibrate an eyepiece graticule and use it to measure the actual size of a specimen. Our fifth learning outcome is to define resolution and magnification and explain the differences between these terms with reference to light and electron microscopy. Let's start with magnification. Magnification is simply how much larger an image appears compared to the actual size of the object. It's a numerical enlargement. However, it's important to understand that high magnification doesn't necessarily reveal more detail. If the image is blurry, making it bigger just makes it a bigger blurry image. This brings us to resolution. Resolution is the ability to distinguish two close points as separate. Think of it as the clarity or detail of an image. A higher resolution means you can see finer details and distinguish between structures that are very close together. Resolution is limited by the wavelength of the radiation used in the microscope. Shorter wavelengths provide higher resolution. Let's summarize the key differences in a table. Magnification is the enlargement of an image, while resolution is the clarity or detail. Magnification determines how big the image looks, while resolution determines how sharp or detailed it is. Magnification depends on the lenses, while resolution depends on the wavelength of light or electrons. The limitation of magnification is that a very high magnification can still give a blurry image if the resolution is poor. The limitation of resolution is that you cannot resolve objects closer than the microscope's resolution limit. We've now defined and differentiated between magnification and resolution, two critical concepts in microscopy. Let's compare light microscopes and electron microscopes based on their features. Light microscopes use light as their radiation source, while electron microscopes use a beam of electrons. As we saw, electron microscopes have a much higher maximum magnification and resolution. This means they can visualize much smaller structures. For example, with a light microscope, you can see the nucleus, cytoplasm, and cell wall. But with an electron microscope, you can see most organelles, including internal structures like ribosomes and membranes, which are too small for a light microscope. There are also differences in the specimen type and preparation. Light microscopes can view living or dead specimens, and preparation is relatively simple and quick. Electron microscopes, however, require the specimen to be dead and placed in a vacuum, and the preparation process is expensive and complex. Light microscopes produce a 2D image, while electron microscopes can produce either 2D TEM or 3D SEM images. Finally, light microscopes are much cheaper and simpler to use compared to the expensive and complex electron microscopes. Let's conclude with the key takeaway. Magnification tells you how big the image is, while resolution tells you how clear the image is. A high magnification is useless without high resolution. You can make an image as big as you want, but if it's not clear, you won't see any more detail. Therefore, resolution is often considered the more important factor in microscopy. We have now thoroughly compared light and electron microscopes and concluded that resolution is key for seeing detail. Now we'll shift our focus from microscopy to the cells themselves. Our next learning outcome is to recognize organelles and other cell structures found in eukaryotic cells and outline their structures and functions. We'll start with the cell surface membrane, also known as the plasma membrane. Its structure is described by the fluid mosaic model. It's made of a phospholipid bilayer with proteins embedded within it. It also contains cholesterol, glycoproteins, and glycolipids. The neoprotein's fluid, part of the model, refers to the fact that the phospholipids can move freely within the layers, giving the membrane flexibility. The mosaic part refers to the proteins, which are scattered throughout the membrane like tiles in a mosaic. Let's look at the key components and their roles. Phospholipids form the bilayer, acting as the primary barrier. Proteins, both intrinsic and extrinsic, are crucial for transporting substances across the membrane, acting as receptors for signals and functioning as enzymes. Cholesterol provides stability to the membrane while also maintaining its fluidity. Glycoproteins and glycolipids, which are carbohydrates attached to proteins or lipids, are involved in cell recognition and signaling, acting like identification tags for the cell. So, we've now recognized the structure of the cell surface membrane and outlined the roles of its key components. Now let's discuss the functions of the cell surface membrane. The cell membrane has several vital roles. First, it controls the entry and exit of substances. It is selectively permeable, meaning it allows some substances to pass through while blocking others. Second, it's involved in cell communication. Receptor proteins on the surface bind to signaling molecules, allowing cells to communicate with each other. Third, it's crucial for cell recognition, especially by the immune system, which identifies cells based on antigens on their glycoproteins. Fourth, it provides structural support to the cell, helping to maintain its shape. And fifth, it forms a clear boundary between the cell's internal environment and the external environment. Here are a couple of additional notes about the cell membrane. First, the cell surface membrane is found in both animal and plant cells. It's a universal feature of all cells. Second, its permeability is not fixed. It can change in response to various signals or conditions, such as temperature. For example, at higher temperatures, the membrane becomes more fluid and permeable. We have now outlined the key functions of the cell surface membrane and noted its presence in all cells and its dynamic nature. Let's move on to the nucleus, the control center of the cell. The nucleus is the largest organelle in eukaryotic cells. It's surrounded by a double membrane called the nuclear envelope. Inside, it contains chromatin, which is a complex of DNA and proteins. It also has one or more dense, dark staining regions called nucleoli. The nucleus has several critical functions. Its primary function is to store DNA, which carries all the genetic instructions for the cell. By controlling gene expression, the nucleus controls all the cell's activities. It is also the site of DNA replication, where the cell makes copies of its DNA, and transcription, where DNA is used as a template to make RNA. Let's look at the nuclear envelope in more detail. The nuclear envelope is a double membrane that completely surrounds the nucleus. It's not a solid barrier, however. It contains numerous nuclear pores, which are channels that allow for the exchange of molecules between the nucleus and the cytoplasm. This regulated transport is essential for the cell's function. We've now recognized the structure of the nucleus and nuclear envelope and outlined their key functions. Let's continue our discussion of the nucleus by focusing on the nucleolus. The nucleolus is a dense, dark staining region found inside the nucleus. It's unique because it is not membrane-bound, unlike most other organelles. It's primarily made of ribosomal RNA, or rRNA, along with DNA and proteins. The nucleolus has a very specific and important function. Its main job is to synthesize ribosomal RNA, or rRNA. It then assembles this rRNA with proteins to form ribosomal subunits. These subunits are then exported from the nucleus through the nuclear pores into the cytoplasm, where they will come together to form functional ribosomes, the sites of protein synthesis. Let's summarize what we've learned about the nucleus and its components. The nucleus is a large, membrane-bound organelle with chromatin that controls cell activities and stores DNA. The nuclear envelope is a double membrane with nuclear pores that separates the nucleus from the cytoplasm and controls transport. The nucleolus is a dense region inside the nucleus that produces rRNA and assembles ribosomes. Together, these structures ensure the cell's genetic information is protected and used correctly. We have now recognized the structure and function of the nucleus, nuclear envelope, and nucleolus. Next, we'll explore the rough endoplasmic reticulum, or RER. The RER is a network of flattened, membrane-bound sacs called cisternae. Its most distinctive feature is that ribosomes are attached to its outer surface, giving it a rough appearance under the microscope. The RER is also connected to the nuclear envelope, forming a continuous membrane system. The RER plays a crucial role in protein synthesis and modification. The ribosomes on the RER synthesize proteins that are destined for export from the cell or for use in membranes. As these proteins are synthesized, they enter the lumen of the RER, where they undergo modification. This can include folding into their correct 3D shape, or having sugars added to them to form glycoproteins. The RER then transports these modified proteins to the Golgi apparatus via small membrane-bound sacs called vesicles. The RER is especially abundant in secretory cells, like those in the pancreas that produce digestive enzymes, or plasma cells that produce antibodies. Let's summarize the key features of the RER. The RER appears as flattened sacs with ribosomes on its surface. Its main function is the synthesis, folding, and transport of proteins. It is continuous with the nuclear envelope and is a key player in the cell's protein production and secretion pathway. We have now recognized the structure of the RER and outlined its functions in protein synthesis and transport. Closely related to the RER is the smooth endoplasmic reticulum, or SER. Like the RER, the SER is a network of membrane-bound tubules and sacs. However, its key structural difference is that it lacks ribosomes on its surface, giving it a smooth appearance. It is often found interconnected with the RER. The SER has a very different set of functions compared to the RER. The SER is primarily involved in the synthesis of lipids, including phospholipids for membranes and steroids like hormones. It also plays a role in the metabolism of carbohydrates. A major function, especially in liver cells, is the detoxification of drugs and poisons. It modifies these harmful substances to make them less toxic and easier to excrete. Finally, the SER is a storage site for calcium ions, which is particularly important in muscle cells, where it's called the sarcoplasmic reticulum and regulates muscle contraction. Let's compare the RER and SER directly. The main difference is the presence of ribosomes. The RER has them, the SER does not. This leads to their different main products. The RER produces proteins, while the SER produces lipids and steroids. Consequently, the RER is common in secretory cells, while the SER is common in cells involved in detoxification, like liver cells, or calcium storage, like muscle cells. We have now recognized the structure of the SER and outlined its functions, and we've clearly differentiated it from the RER. Our next organelle is the Golgi apparatus, also known as the Golgi body or Golgi complex. The Golgi apparatus is a stack of flattened, membrane-bound sacs, which are also called cisternae. It's often located near the endoplasmic reticulum and the nucleus. A key structural feature is that it has two distinct faces: the cis face, which is the receiving side that faces the ER, and the trans face, which is the shipping side that faces the plasma membrane. You'll also see vesicles budding off from its edges. The Golgi apparatus is the cell's post office, modifying, sorting, and packaging molecules. Its first function is to modify proteins received from the RER. For example, it can add sugars to proteins to make glycoproteins. Second, it packages and sorts these modified proteins into vesicles. These vesicles can then be used for secretion, a process called exocytosis, or for delivery to other organelles or the plasma membrane. Third, the Golgi produces lysosomes, which are vesicles containing digestive enzymes. And fourth, in plant cells, it synthesizes certain polysaccharides for the cell wall. Let's look at some examples of products that come from the Golgi. Examples of Golgi products include enzymes, such as the digestive enzymes found in lysosomes, glycoproteins, like antibodies, which are crucial for the immune system, and secretory proteins, like insulin, which is a hormone secreted by pancreatic cells. These examples highlight the diverse and vital roles of the Golgi apparatus. We have now recognized the structure of the Golgi apparatus and outlined its functions in modifying, sorting, and packaging proteins and lipids. Next up are the mitochondria, often called the powerhouses of the cell. Mitochondria are double-membrane organelles. The outer membrane is smooth and controls the entry and exit of substances. The inner membrane is highly folded into structures called cristae. These folds are crucial because they greatly increase the surface area for chemical reactions. The fluid-filled central space inside the inner membrane is called the matrix. The matrix contains enzymes for respiration, small circular DNA, and 70S ribosomes. The primary function of mitochondria is energy production. Mitochondria are the site of aerobic respiration, the process that produces ATP, which is the energy currency of a cell. The cristae provide a large surface area for the enzymes involved in ATP production, such as those in the electron transport chain. The enzymes in the matrix catalyze the Krebs cycle, another key stage of respiration. A unique feature is the mitochondrial DNA, which allows mitochondria to replicate independently of the cell nucleus and synthesize some of their own proteins. The presence of circular DNA and 70S ribosomes in mitochondria is significant. It strongly supports the endosymbiotic theory, which proposes that mitochondria evolved from free-living prokaryotes that were engulfed by early eukaryotic cells. This theory explains why mitochondria have their own genetic material and ribosomes, similar to bacteria, and can produce some proteins and enzymes independently of the nucleus. We have now recognized the structure of mitochondria and outlined their functions in energy production and their unique features. Let's now discuss ribosomes, the cell's protein factories. Ribosomes are small, dense structures made of ribosomal RNA, or rRNA, and proteins. They are unique because they are non-membrane-bound organelles. Each ribosome is composed of two subunits, a large one and a small one, which come together to perform their function. There are two main types of ribosomes in eukaryotic cells. The first type is ADS ribosomes. These are larger and are found free in the cytoplasm or bound to the rough endoplasmic reticulum. Free ribosomes synthesize proteins that are used inside the cell, while ribosomes on the RER synthesize proteins that are exported from the cell or used in membranes. The second type is 70S ribosomes. These are smaller, like prokaryotic ribosomes, and are found exclusively in mitochondria and chloroplasts. They synthesize proteins specifically for the function of these organelles. The presence of 70S ribosomes is another piece of evidence for the endosymbiotic theory. Just like the circular DNA, the presence of 70S ribosomes in mitochondria and chloroplasts supports the idea that these organelles were once free-living prokaryotes. It's also important to note that ribosomes are too small to be seen under a light microscope, but they are clearly visible in electron micrographs. We have now recognized the structure of ribosomes and outlined their functions in protein synthesis, differentiating between the two types found in eukaryotic cells. Let's now discuss lysosomes, the cell's recycling centers. Lysosomes are spherical, membrane-bound vesicles that are formed by the Golgi apparatus. They contain a variety of powerful hydrolytic or digestive enzymes, such as proteases, lipases, and nucleases. These enzymes are kept safely enclosed by a single membrane, preventing them from digesting the cell itself. Lysosomes are involved in several types of intracellular digestion. First, they perform intracellular digestion of worn-out organelles, a process called autophagy. They engulf and break down old mitochondria or other organelles, recycling their components. Second, they digest ingested materials, like bacteria, that have been taken into the cell through phagocytosis. This is especially important in immune cells. Third, they are involved in autolysis, which is programmed cell self-destruction. This is a controlled process that is essential for development and tissue turnover. Finally, they play a role in the immune response by digesting foreign substances. Here are some important notes about lysosomes. Lysosomes are mostly found in animal cells and are rare in plant cells. Their enzymes work best at an acidic pH, which is maintained inside the lysosome. They are sometimes called suicide bags of the cell due to their role in autolysis. And as we mentioned, they are formed by budding off from the Golgi apparatus. We have now recognized the structure of lysosomes and outlined their functions in intracellular digestion and recycling. Finally, let's discuss centrioles and microtubules, which are key components of the cytoskeleton. Centrioles are small, cylindrical structures composed of microtubules. They are typically found in pairs, positioned at right angles to each other. Each centriole is made of 9 triplets of microtubules arranged in a circular pattern. often called a 9x3 structure. They are located in the centrosome, which is the microtubule organizing center near the nucleus. Centrioles have several important functions, especially during cell division. Their primary role is in organizing the mitotic spindle during cell division. They help in the formation of the spindle fibers, which are made of microtubules that move chromosomes to opposite ends of the cell. They also play a role in the formation of cilia and flagella, which are hair-like structures involved in cell movement. Now let's look at microtubules themselves. Microtubules are long, hollow protein tubes made of a protein called tubulin. They are a major component of the cytoskeleton, the cell's internal scaffolding. They are dynamic structures, meaning they can grow and shrink as needed, allowing the cell to change shape and move things around. We have now recognized the structure of centrioles and microtubules and outlined their functions in cell division and support. Let's continue with the functions of microtubules and then compare them to centrioles. Microtubules have several key functions. First, they provide mechanical support and help maintain cell shape. Second, they form the spindle apparatus during mitosis and meiosis, just as we saw with centrioles. Third, they act as tracks for the movement of organelles and vesicles inside cells, like secretory vesicles moving from the Golgi to the plasma membrane. Fourth, in cilia and flagella, they are arranged in a characteristic 9+2 pattern, which enables these structures to move. Let's compare centrioles and microtubules. Centrioles are composed of 9 triplets of microtubules, while microtubules are made of tubulin protein. Centrioles are located near the nucleus in the centrosome, while microtubules are found throughout the cytoplasm. The main function of centrioles is to organize the spindle during cell division, while microtubules support cell shape, transport, and movement. Centrioles are present in animal cells, but absent in most higher plant cells, whereas microtubules are found in all eukaryotic cells. We have now outlined the functions of microtubules and compared them to centrioles, completing our survey of key eukaryotic organelles. Let's quickly recap the key concepts we've covered in this video. We started by learning how to prepare temporary mounts for microscopy, emphasizing the importance of thin specimens, stains, and careful cover slip placement. We then discussed the purpose and guidelines for creating accurate biological drawings. We learned how to calculate magnification and actual size using a simple formula and scale bars. We differentiated between magnification, which is how big an image is, and resolution, which is how clear it is, and compared light and electron microscopes. Finally, we explored the structure and function of several key eukaryotic organelles: the cell surface membrane, nucleus, nuclear envelope, nucleolus, rough and smooth endoplasmic reticulum, Golgi apparatus, mitochondria, ribosomes, lysosomes, centrioles, and microtubules. You should now be able to make temporary preparations, draw cells accurately, perform magnification calculations, understand the difference between magnification and resolution, and identify the structure and function of the organelles we discussed. Thank you for watching.